The world of chemistry is ever evolving. Scientists are constantly pushing boundaries, seeking new compounds and reactions that can benefit humanity. One such discovery is hexahydrocannabinolic acid, or HHCA. This novel cannabinoid is synthesized through a process called hydrogenation. HHCA holds immense potential, not just in the realm of synthetic chemistry, but also for its biological activity. The discovery of HHCA marks a significant step in cannabinoid research. From its origins in the lab to its potential impact on human health, HHCA represents a new frontier in chemistry. Hydrogenation is a fundamental process in chemistry. It involves the addition of hydrogen molecules to a substance. This seemingly simple act can dramatically alter a compound's properties, creating new possibilities. By saturating the double bonds present in cannabinoids, we can create new molecules with enhanced stability and potentially improved therapeutic benefits. The transformation of THC to HHCA exemplifies the power of hydrogenation. What was once THC, known for its psychoactive effects, becomes HHCA, a molecule with potentially different therapeutic applications. By harnessing the power of this chemical reaction, we can unlock new possibilities in the development of novel therapeutics. Let's delve into the heart of hydrogenation. The double bond. Imagine two carbon atoms, the building blocks of organic molecules, linked together. A double bond involves two shared pairs of electrons. These double bonds are like busy intersections in a molecule, sites of potential reactivity. Hydrogenation comes into play by adding hydrogen atoms to these double bond intersections. The double bond becomes a single bond, now sharing only one pair. In the world of cannabinoids, this saturation has profound effects. THC, the psychoactive component of cannabis, owes its properties to a specific double bond in its structure. Hydrogenation removes this double bond, transforming THC into HHCA. Cannabis is a complex plant. It possesses a rich tapestry of chemical compounds, each contributing to its overall effects, while THC and CBD often steal the spotlight. A symphony of other molecules, including terpenes and flavonoids, play a crucial role. This intricate interplay of compounds is known as the ensemble effect. Terpenes, for example, are aromatic molecules responsible for the distinct scents of different cannabis strains. They also interact with cannabinoids, influencing their absorption and effects. By saturating double bonds in various cannabis compounds, we alter their individual properties and potentially enhance their synergistic effects. This opens up exciting possibilities for tailoring cannabis-based therapies. The path to scientific discovery is rarely linear. It often winds through unexpected landscapes, connecting seemingly disparate fields. Such is the case with the journey that led to the hydrogenation of cannabinoids. It began, surprisingly, with catnip. Catnip contains a molecule called nipetolactone, structurally similar to some cannabinoids. Chemists discovered that hydrogenating catnip oil could enhance its insect-repelling properties. This finding sparked a question. Could hydrogenation be applied to other plant-derived compounds, like cannabinoids, with similar success? Nature, in all its wisdom, has provided us with a treasure trove of medicinal plants. Cannabis, with its complex array of compounds, is a prime example. Modern science allows us to peer into the molecular world of plants, unraveling the secrets behind their healing powers. One key insight from studying cannabis is the entourage effect, where various compounds work together in synergy. Hydrogenation offers a way to mimic and potentially enhance nature's design. By saturating double bonds in cannabinoids, we can fine-tune their properties and potentially amplify their synergistic effects. This technology could unlock new frontiers in cannabis-based medicine. Let's zoom in on a specific example of hydrogenation's transformative power, the conversion of THC to hexahydrocannabinol, or HHC. THC, with its iconic double bond structure, is known for its psychoactive effects. When we introduce hydrogen, breaking that double bond and saturating the molecule, we create HHC, a compound with a distinct chemical identity. HHC, while structurally similar to THC, exhibits a different pharmacological profile. Its interaction with the body's endocannabinoid system is distinct from that of THC. HHC could hold promise for therapeutic applications where THC may not be suitable. This example highlights the power of hydrogenation to unlock the hidden potential within cannabinoids. The journey from THC to HHC is a testament to the ingenuity of synthetic chemistry. Returning to the concept of the entourage effect, let's imagine the cannabis plant as a grand orchestra. Each compound, from the prominent THC and CBD to the subtler terpenes and flavonoids, plays a distinct role in creating the overall symphony of effects. Now envision hydrogenation as a skilled conductor, able to fine-tune the individual instruments and harmonize their contributions. By selectively saturating double bonds in specific compounds, we can potentially enhance certain notes, soften others, and create a more balanced and therapeutic composition. This analogy underscores the potential of hydrogenation to not only create novel cannabinoids like HHC, but also to enhance the synergistic effects of existing compounds. This vision of a fine-tuned cannabis orchestra highlights the exciting future of this technology. Sometimes, the most unexpected places hold the key to scientific breakthroughs. Take catnip, for instance. This unassuming herb, known for its intoxicating effect on felines, played a pivotal role in the journey toward hydrogenated cannabinoids. Hydrogenating catnip oil, which contains nepentolactone, significantly enhanced its ability to repel insects. This discovery sparked a wave of research, with scientists eager to explore the potential of hydrogenating various natural products. Among those intrigued were researchers studying cannabinoids, the fascinating group of compounds found in cannabis. The lessons learned from catnip underscore the interconnectedness of the natural world and the power of scientific curiosity. The journey from a scientific discovery in the lab to a widely available product is often long and arduous. The development of hydrogenated cannabis oil, or HCO, is a prime example of this complex process. Initially, the production of HCO was confined to small-scale, laboratory settings. However, as the potential benefits of HCO became increasingly apparent, the need for larger-scale production methods became paramount. This shift required significant investment in specialized equipment and expertise. Large-scale hydrogenation reactors had to be designed and constructed. Despite these challenges, the allure of HCO's potential therapeutic benefits fueled the drive for industrial-scale production. The unveiling of HCO as a new frontier in cannabis science has sparked immense excitement and a flurry of research activity. Scientists are eager to unravel the full therapeutic potential of this novel compound. Early findings suggest that HCO may possess a unique pharmacological profile, distinct from its non-hydrogenated counterparts. One area of particular interest is HCO's interaction with the endocannabinoid system, or ECS, preliminary studies suggest that HCO may interact with the ECS in novel ways, potentially leading to therapeutic benefits not seen with traditional cannabinoids. Researchers are particularly interested in HCO's potential as an anti-inflammatory, analgesic, and neuroprotective agent. The initial findings are promising, suggesting that HCO may represent a valuable addition to the growing arsenal of cannabis-based therapies. Glioblastoma multiforme. This devastating brain cancer, known for its rapid growth and resistance to conventional treatments, leaves patients and families grappling with a bleak prognosis. Amidst this ongoing battle, a glimmer of hope emerges from the world of cannabinoid research. Hydrogenated cannabinoids, particularly HHC, have shown promise in preclinical studies, exhibiting potential anti-cancer activity against glioblastoma cells. One key mechanism by which HHC exerts its anti-cancer effects is its ability to inhibit angiogenesis, the formation of new blood vessels that tumors rely on for growth and survival. By cutting off the tumor's blood supply, HHC effectively starves it, slowing its progression and potentially shrinking its size. While rigorous clinical trials are needed to confirm these preclinical findings in humans, the initial results offer a compelling reason for optimism. The discovery of HHC's potential anti-cancer activity against glioblastoma has opened up a Pandora's box of questions. How does this hydrogenated cannabinoid exert its effects? What molecular pathways are involved? One promising lead lies in HHC's interaction with the endocannabinoid system, a complex network of receptors and signaling molecules that plays a crucial role in regulating cell growth, proliferation, and survival. Research suggests that HHC may interact with specific receptors within the ECS, modulating their activity and influencing downstream signaling pathways involved in tumor growth and progression. One potential target is the CB2 receptor, which is often overexpressed in glioblastoma cells. By activating the CB2 receptor, HHC may trigger a cascade of events that ultimately lead to tumor cell death. This ongoing quest to understand the intricate molecular dance between HHC and the ECS is crucial for developing more targeted and effective therapies for glioblastoma and potentially other types of cancer as well. Section 3. The Metabolism of HHC, a unique profile. The journey of a drug through the human body involves a series of chemical transformations. Understanding drug metabolism is crucial for assessing safety and efficacy. HHC undergoes metabolic transformations, creating unique metabolites. Oxidation, carried out by liver enzymes, is a key pathway. Cytochrome P450 enzymes convert HHC into water-soluble forms. These metabolites have distinct structures and activities. Some metabolites contribute to therapeutic effects, others may be inactive or toxic. Understanding HHC metabolism helps optimize therapeutic potential and minimize side effects. This knowledge aids in designing effective dosing regimens and novel HHC analogs. The promise of hydrogenated cannabinoids. Hydrogenated cannabinoids like HHC show unique properties and therapeutic potential. Transforming THC to HHC demonstrates hydrogenation's power, offering new therapeutic applications. HHC shows promise in combating aggressive cancers like glioblastoma. It may inhibit angiogenesis, starving tumors of their blood supply. Rigorous clinical trials are needed, but the potential benefits warrant continued exploration. A new horizon in cannabis science. Hydrogenated cannabinoids mark a pivotal moment in cannabis science. This shift allows creating novel molecules with tailored properties. Fine-tuning cannabinoids opens up new possibilities for cannabis-based therapies. Imagine personalized cannabis medicine as reality. Hydrogenation could create tailored extracts for individual needs. This precision could revolutionize healthcare with targeted treatments.